Aging is written into our cells, not just in their genes, but in their shape, texture, and behavior. Under a microscope, a senescent cell practically shouts its age. It's larger, flatter, often riddled with vacuoles and brimming with granularity. But as obvious as these features might look to a trained human eye, they're still subjective, slow to assess, and prone to inconsistency. Enter machine vision, an AI-powered transformation in how we see cellular aging, quite literally. With deep learning algorithms now able to detect the faint, intricate signatures of senescence, science is on the cusp of diagnosing, quantifying, and even predicting aging with unprecedented precision. Machine vision is essentially what happens when you give a microscope eyes and a brain. These systems combine high-resolution imaging with neural networks trained to recognize minute visual differences that might be invisible or subtle to humans. For senescence detection, this turns out to be a perfect match. Senescent cells across tissues display telltale morphological signs: altered nuclear size, cytoplasmic expansion, chromatin condensation, mitochondrial bloating, and changes in cell border definition. While any one of these features might hint at aging, it's the combination, the full visual fingerprint, that gives the cell away. And that's where computer vision excels: pattern recognition at scale. These algorithms, once trained on labeled datasets of senescent and non-senescent cells, can process thousands, sometimes millions, of cell images, rapidly classifying them with a level of granularity no human technician could replicate. What's more, they're objective. Unlike manual scoring, which can vary between researchers or labs, machine vision applies a consistent standard, ensuring reproducibility and reducing bias. It's like having a tireless, hyper-focused cytologist with superhuman recall and no coffee breaks. But the magic goes beyond just recognition. Machine vision systems are increasingly being used to uncover novel phenotypes of senescence, cell states that fall outside traditional definitions. For instance, some senescent cells may appear normal in size, but have nuclear envelope blebbing or subtle mitochondrial clustering patterns. Others may show early cytoskeletal reorganization long before P16 or SS galactosidase show up. By training on massive image libraries across tissues, skin, lung, liver, heart, AI is beginning to identify these nuanced, tissue-specific forms of senescence, helping researchers understand how aging manifests differently across the body. This leads to the tantalizing possibility of creating a visual atlas of cellular aging, a map of senescent morphologies across cell types, age ranges, disease states, and even interventions. Want to know how senescent kidney cells look in diabetic nephropathy? Or how cardiac fibroblasts change after chemotherapy? Machine vision can show you, often picking up on morphometric details like nuclear roundness, membrane texture, or autofluorescent pigment distribution that correlate with function or pathology. Beyond basic research, the clinical implications are immense. Imagine a biopsy where AI doesn't just scan for cancer, but evaluates the senescent burden of the tissue, predicting organ resilience, regenerative capacity, or response to therapy. In regenerative medicine, where senescent cells can hinder stem cell grafts or tissue engineering, real-time imaging tools guided by machine learning could ensure senescence-free constructs. In drug development, AI-driven screening platforms can evaluate how experimental compounds alter senescent morphology across different cell types, shortening the time from hypothesis to insight. There's also a growing interest in combining machine vision with multimodal data, integrating visual features with transcriptomic, proteomic, or metabolic readouts. This would allow researchers to connect what a senescent cell looks like to what it's actually doing. For instance, a certain nuclear shape might correspond to a highly inflammatory SESB profile, while another morphological subtype might be metabolically inert. This fusion of data could finally allow us to distinguish harmful, zombie, senescent cells from harmless or even protective ones, something current biomarkers struggle to do reliably. And while the lab is where this revolution began, it's not where it will end. Smartphone-based microscopes, AI-assisted dermatology tools, even real-time senescence monitoring and wearable biosensors are all being explored. One day, your doctor may not just check your cholesterol, they may scan a drop of your blood and, with the help of machine vision, tell you how old your cells think you are. So yes, machine vision is changing how we see aging by teaching us to look beyond the obvious. In doing so, it's reframing cellular senescence not as a static fate, but as a dynamic, observable process we can quantify, classify, and eventually manipulate. Because aging isn't just a number, it's a shape, a texture, a pattern. And finally, thanks to AI, we're learning how to read it.